Good morning, everybody. My name is Kirsten Poore. I'm from the University of Southampton in the UK. I do a lot of medical education, so it's a great pleasure to see you here as medical students learning about this very important topic. So I'd like to tell you about a research area that we call developmental origins of health and disease, and I'm also going to be talking about the process of epigenetics. So I want you to be able to understand how those environmental challenges in early life may predispose to later disease. And I'm going to briefly introduce the concept of epigenetics to you. I can't see my next slide. It's there, but on here, I think it's the presentation mode. Yeah. Doesn't match. Can we? Oh, that's better. Thank you. So we all want to think about what makes us healthy. Why is it that some people have more health than others, particularly as we age? And so we all can think about, yes, we should eat a healthy diet, we should do more exercise, but why doesn't that always work? Why are we not all as healthy, given that what we know about health? And what we're particularly interested in these days is that the fact that non-communicable diseases are increasing. And by NCDs, I mean cardiovascular disease, some lung diseases, diabetes and obesity, some neurodevelopmental problems, for example. We know that up to 1 in 8 people in the world a rate of obesity is increasing. We know that the number of people who have diabetes is also increasing rapidly. And so why is that? In developed countries like in France and the UK, in Japan, we know that we eat too much and we don't move enough. So we have some possible causes there. In low and middle income countries, we know that the number of deaths from communicable diseases is falling, but the number of people dying from non-communicable diseases is increasing. And this is in times of socio-economic and nutritional transition. And those countries often have a double burden of both undernutrition and obesity. There are plenty of initiatives to try and improve our health. In the UK, for example, we have various initiatives about eating well and moving more. And there are international efforts to improve things like childhood obesity from the World Health Organization. But they're not always working. And in fact, the non-communicable diseases continue to increase, and in fact, they're the world's biggest killers. And the speed of that change in the increase in diseases cannot be explained by genetic variation. It's not our genes that are changing in this very short space of time over the last couple of decades. So what might it be? What we're particularly interested in, and what we're going to be hearing about in the next 2 days, is that the environment is influencing our risk of non-communicable disease. And we also ask, when in the life course is that environment having the most impact? So we're going to be talking about that in the next couple of days. For us, in our research area, we believe that building a healthy baby is the foundation to lifelong health. And this is the research area that we call developmental origins of health and disease. And this comes from some pioneering work out of the University of Southampton from Professor David Barker. He was able to follow up a series of men who had been born in the 1920s and 30s. And when they were in their 60s, he looked at their health outcomes. And he was able to show that as birth weight of those individuals decreased, so did their rate of cardiovascular disease and their chance of getting diabetes increased. So smaller babies were more at risk of having heart disease and diabetes in much later life. And it's not just in the extremes of birth weight, in very small babies, but it's a graded phenomenon across the birth weight range. So the very early work was particularly interested in the effect of maternal nutrition, and there were a series of studies that looked at people who had been exposed to famine during the Second World War, in which there was a period when the nutritional environment was drastically reduced in the Netherlands, and this included for people who were for women who were pregnant at the time. So there were babies that were exposed to famine in utero at different time points in pregnancy. And this had an effect on the size of babies, in babies that were exposed to famine, and particularly in babies that were exposed to famine in either mid or late pregnancy. They were born smaller than expected because of the lack of nutrition. So what the research teams did was to try and follow up these individuals and look at their risk of non-communicable diseases in later life. And they showed that babies that were exposed to famine in late gestation, who had a reduction in birth weight, had an increased risk of getting diabetes, type 2 diabetes, in later life. They also showed that babies that were exposed to famine in early pregnancy had an increased risk of heart disease in later life. So there's something about the fetal and very early environment that was increasing risk of disease. But it wasn't just birth weight that was the main issue, because some of these babies were not born small. They weren't small when they were born, but they still had an increased risk of heart disease later in life. So birth weight is certainly a measure of the fetal experience, but it's not the only measure that's important. And so we very much like to take a life course approach in how our risk of disease in later life is determined from a very early point from the embryo, and we need to think about our risk of disease over the course of our lives. And this is the concept of DOHAD, Developmental Origins of Health and Disease, that disease risk can be induced by aspects of the environment that's detected very early in the embryo, the fetus and the young child, and affects our healthy ageing. So this is about a concept called developmental plasticity. So when we are very young, we have the ability to respond to the environment, and so our genetic potential will be influenced by the prevailing environment. So as we age, we become less plastic, less able to respond to the environment, and our risk of disease increases over the course of the life course, because we have inadequate responses to new environmental challenges that may come along. And this again occurs across the graded range of normal development. We're not talking about just extremes of environmental exposure. So when we're talking about developmental origins of health and disease, we used to talk about the competition between nature, which is the fetal genome, and nurture, which is the early environment, leading to the fetal developmental period. But we now think about nature being influenced by nurture, and there's an interplay between those two. And that is going to influence our later risk of disease. And on top of that, of course, you have the postnatal environmental environment and lifestyle that can influence risk of disease. And the reason we worry about fetal development particularly is, of course, during fetal development, there are critical windows of development where specific organs and systems are being developed at different times during the developmental period. So depending on the challenge at different time points in that developmental period may determine the later and long-term effect. Some of those effects may be very adverse for the fetus. There may be specific toxic effects that actually cause chromosome abnormalities or birth defects, or in fact fetal death. But other effects of the environment may be not so obvious at the time of birth. It may lead to some changes in growth and maturation and physiology of the fetus, which we don't immediately see at birth. There may be some shorter-term effects that affect fetal growth or may in fact induce premature delivery. And those things, of course, do have an impact on public health. But there may be other much longer-term effects for example, on reproductive capacity or neurological development, or some risk of cancer, and of course non-communicable disease risk that we don't see at the time of birth. They may be more subtle and they appear later in the life course. And we know that the fetus is able to respond to an adverse environment. And Professor Barker and colleague Nick Hales came up with this hypothesis called the thrifty phenotype hypothesis, that the fetus can make irreversible changes in its development that predispose to later disease. For example, in a poor nutrient environment in fetal life, the fetus adapts to respond to that poor environment. And if the postnatal environment is very different to that, that may lead to disease risk. My colleague Mark Hansen from Southampton and colleague Peter Gluck from New Zealand then came up with a different theory about the predictive adaptive response, whereby a fetus may make a prediction about the postnatal environment based on its current fetal environment, and it makes some adaptations to that. And it's whether that postnatal environment matches the prenatal prediction is whether that individual may be suited to the environment or may be increased risk of disease. And so the fetus is quite able to sense the external environment, and it will be able to respond to challenges and exposures depending on the stage of pregnancy and other physiological and pharmacokinetic changes that are taking place during pregnancy. So it can make changes and adaptations to that external environment. We now know from many years of research that a whole range of tissues may be involved. For example, in the risk of adult metabolic disease, there are a whole range of potential perturbations or changes in the early life environment, such as nutrient restriction or gestational diabetes or maternal obesity, that affects a whole range of fetal tissues that go on to increase the risk of glucose intolerance, diabetes type 2 risk in later life. Tissues from adipose tissue, pancreas, kidney, brain, skeletal muscle, all have observable effects within them that may be contributing to this disease risk. So what do we really mean by the environment? I've been talking about nutrition, which is my particular research interest, and other maternal factors, such as mother's body composition and age, stress and lifestyle factors. But what we're also going to be particularly talking about in the next couple of days is other external factors, chemicals, toxicants, pollutants, and you're going to be hearing a lot more about that in the next 2 days. And there are a lot of chemicals in our everyday environment. They're in just about everything we touch and see and breathe, and you'll hear about them. But the important thing is that many of those potential toxicants have the ability to cross the placenta and influence fetal development. You're also going to be hearing about climate change and how climate change also has an effect on our basic health requirements. And this will affect both adults, but of course vulnerable people like pregnant women and young children. And Dr. Etzel is going to be telling you more about that in due course. And so it's important to remember that these environmental challenges don't exist in isolation. And at any one time, we may be exposed to poor nutrition, the effects of climate change, toxicants, air pollution. All of these things will be a big mix of the environment that could be affecting fetal development. So what actually brings about the changes in the fetus and the early infant? What is having this actual effect to bring about risk of disease? Well, the process of epigenetics is the study of heritable changes in gene function that do not involve any change in the actual DNA sequence. So we know that the DNA sequence is not going to change, but the epigenetic processes around that DNA determine how those genes actually function. And it's this that brings phenotype to life, really, and it also is a process that enables developmental plasticity. So it's the interaction of the environment on our genes. There are 3 different types of epigenetic processes. There are changes in the histone proteins that alter chromatin structure of the DNA. There are changes in microRNAs. These are non-coding RNAs that are not translated into proteins, but can be associated with gene silencing. And there's also evidence of changes in methylation patterns on the DNA in certain promoter regions of different genes. So these are particularly putative mechanisms whereby the environment may be changing the exposure, the expression of different genes. And some of these changes may be permanent or transient. They are a part of normal development, but can be affected by the environment. And that's what, in fact, leads to different phenotypes, different cell types making different decisions through the developmental pathways. And it can lead to things like the difference between a queen bee and a worker bee, depending on the sort of environmental diet of the bee larva triggers a switch which determines the actual phenotype. It's also why the very first cat that was cloned looks nothing like its mother, because of epigenetic processes have changed the phenotype of that clone, even though they have the exact same DNA sequence. We know that the methylation patterns are changed in very early life. There's a process of resetting methylation patterns from our early primordial cells through to the zygote and through to the embryo. There's a wave of epigenetic methylation changes. And we think that specifically in this very early period that the environment can influence the changes in those methylation patterns. So the epigenetics really gives a memory of development because it responds to the environment at that time. There's examples of how maternal behaviour can in fact induce changes in epigenetics in the offspring. This is looking at the glucocorticoid receptor in the hippocampus, which is part of our stress response. And this is an animal study looking at rats and whether the mother rats were very good looking after their pups or whether they were poorer mothers. And the poorer mothers, so the rats that had less attention to their offspring, actually had differences in the methylation of that glucocorticoid receptor in later life, and that changed how they responded to stress. So an example of how maternal behaviour can influence epigenetics. And we also know that childhood body composition can be influenced by the epigenetic state at birth. These are a couple of studies from Southampton which looked at the methylation pattern of a particular gene, RXRA, which is a transcription factor known to be involved in insulin sensitivity. And they found that only a 20% change in variance, the variance in body composition was explained by 20% by the methylation status of RXRA. So this one particular epigenetic change was influencing the body composition of children aged 6 and 9. And this opens quite a good avenue for potential biomarkers where we may be able to predict later risk of disease if we could measure some of those epigenetic changes very early in life. So we're all about thinking about developmental match or mismatch, and the epigenetic changes in early life help to tune the phenotype to match the prevailing environment. So it's sensing the environment and making appropriate changes. But if those changes are not appropriate, it may lead to increased risk of disease if the environment later in life does not match what was predicted. So today and tomorrow we're going to be talking about a lot of external environmental factors, and as I said, many of these are known to cross the placenta, and you'll be hearing about how they have demonstrated or highly suspected effects on the risk of disease later in life. And it's epigenetic processes that are involved in that. There's increasing evidence now that effects of these environmental toxicants are causing epigenetic changes that are visible in later life. I've talked a lot about the effect on pregnancy and on mothers, but we also need to think about the paternal environment. There is increasing research in this area now that the father's environment can influence sperm development and thereby induce risk of disease in their offspring. And there is some data here showing that the exposure of chemicals in certain occupations of fathers is linked to certain fetal abnormalities or risk of later disease. And we now know that there's evidence for paternal diet influencing epigenetic changes and influences in the sperm, which can affect the developing fetus. So something to also think about the role of the father here. So impairment of paternal genes and the epigenome in the father will also be required. It may also increase risk of poor development in the fetus. So if we have a life course view of disease risk whereby it increases over as we age, what could we do about this? Could we intervene? Could we improve our risk of disease with this knowledge? If it increases with no intervention, certainly if we make some interventions like healthy diet or exercise in later life, we could reduce our risk of disease. But if we make an intervention earlier in life, in childhood for example, we can change that trajectory. If we make an intervention even earlier in life, during pregnancy and with very young children, we can have a potentially much greater impact on the risk of disease, reducing it more than we might have if you just have an intervention in later life. So the message really is about improving our environment, and it can help shape the health of our future generations, help increase our chance of having a healthy aging. But it's not going to be easy. There are particular challenges in this area of research. As I said, some of the developmental effects may not always be apparent at birth, so how can we predict which individuals might go on to be more at risk of disease? And some very small effects may become much bigger in later life, or the response might change. Some effects of the early environment may not be reversible, and we have to be aware of that, depending on when the challenge occurred in particular critical periods of development. There's increasing evidence that some of these changes may actually be transmitted down the generations. There are multi-generational effects of exposures several years ago. One of the things we particularly would want to do is to be able to identify at-risk individuals. If we can find people that may be more at risk of disease later in life, we may be able to intervene sooner. But ideally, of course, we want to be able to improve the environment for parents and children, but that is sometimes very complex. As future healthcare professionals, we believe that you have a role here to play. By coming to this course, you will be aware now of this area of research, and so that's going to hopefully influence your practice as clinicians. We would ask that you support avoidance of exposures in your practice, and you can advocate for your patients. You can support those patients to have healthy lifestyles for themselves and for their children and future generations. So we'll just finish with this quote from Frederick Douglass, a very long time ago, but absolutely important still today. It's easier to build strong children than to repair broken men. So thank you very much.
Thank you very much, Kirsten. So next speaker will be Professor Ruth Etzel. And after Professor Ruth Etzel's presentation, there will be discussions. Now we take a... Okay. And any question or comments from students?
Well, this is... You can do both. Yes, questions. Questions. Allez, allez. Maybe I can start. Can I start to ask a question? Okay, you mentioned that now with epigenetic profile, we can have some idea about the exposure. You know that we can do a full genome sequencing now, and we can get some, ideas about your susceptibility to maybe disease. Do you think it's worthwhile for every new child, I mean, to have an epigenetic profile to guess what will happen later on?
That's a challenging question, isn't it? Because yes, in theory, it might be possible. Would you want to know your entire genetic?
If I can prevent something, I mean.
If you can prevent something, I think it's an ethical question as well as a scientific question, because some people may not want to know everything. But I do think there are certain things that we might want to definitely be able to predict for people.
Yeah. Students, questions? Come on. Yes. Okay.
Okay, thanks for your presentation. I'm a student come from the Chiba University. And my question is, my major is try to deal with the pediatric cancers. And in the pediatric cancer, the famous cancer is the neuroblastoma, which is led by the MECAN genes amplifications, which is a famous epigenetics questions. And my question is that, like the environment challenging or some pollution will have any relation to this non-random non-random epigenetic changes, or it's just related with all random epigenetic changes. That's all. Thank you.
Is it related to non-random?
Yeah. Sorry, I'm wondering that the environment changes will related to the non-random epigenetic changes, like the gene amplifications, or some the chromosome translocations, like the will lead to some cancer fusion genes.
I don't know that specific case, but I think it's all possible. I'd have to look that one.
I think most people in the field believe that cancers of children start actually in the fetus. I mean, it's the susceptibility, and it is likely to be some kind of epigenetic change. But this is, I mean, work is still going on in this field. It's difficult, you know, because luckily children cancer are rare. So if you want to do a really good study, you need a lot of observation, a big cohort of people. So it's a little bit tricky. There's currently in France a big cohort that will start that aims at better identifying the mechanism of these children cancer. D'autres questions? Où est-ce qu'il y a?
I've put,
okay,
there's another place, if you want to ask questions anonymously, not out loud, you can put a question into this particular Vivox app, and we will see the question and we can feed it to the presenters as we go through. That will be open throughout today and tomorrow.
I'm the student of the Chiba University, and I'm studying about the bacteria in the intestinal. So I'm very interested in if we eat some food or something to make the intestinal bacteria change, and maybe it can make people be healthier or not.
Yeah, there's a huge amount of research now about the gut microbiota. And that's very important because, of course, we are born with a set amount of bacteria in our guts. And even the way that we are born changes that. A vaginal delivery versus a cesarean section, a baby will have different gut bacteria. And so that will certainly be influenced by the very early environment. And the risk of disease changes over the life course and can be attributed to different populations of gut bacteria. It's a very active area of research, very important.
Okay, thank you very much. And I have another question about some children have ADHD or some ASD, like this problem. Some people think it's because the environmental is right or not.
Yeah, there is certainly evidence for that, yes. Brain development, of course, very, very important in fetal life and in early postnatal life. So there is a lot of evidence now that some environmental challenges are increasing the risk of ADHD and autism.
Thank you very much.
So we have 2 for Chiba and 0 for Nice. Allez, les Niçois, là. Questions, again? Okay, well, over there, yeah, voilà.
Hi, I'm a student from Nice. We know that most pollutants are omnipresent in our environments, and there's not much as an individual we can do about it. So on the practical side, as a mother, for instance, who is having a child, what can she do to avoid these? I mean, it seems like it's kind of how can you say that? There's nothing to avoid it, you know? So I think it's something more like on the governments or the bigger picture. I mean, as an individual, we can't do about it. And as doctors, apart from being part of government associations or stuff like that, we can't do much about it. So what could we do for it?
I understand that. There are some things that we can't control for at a government level, but there are individual choices that people and mothers can make about the exposures in their household, with their diet. I think Patrick wants to, because there is going to be a session later about
he's
prevention.
giving a talk about it.
Tomorrow, particularly about prevention.
Pregnant woman patient to give practical advice. We'll have a talk tomorrow on this subject. We will answer to your question. Very important question. Thank you.
Okay, other questions?
Yes.
Yes, here.
Yes, Ebiko.
Yeah, so you spoke about biomarkers which can predict some disease. So in your university, do you use actually some biomarkers in practice, or is it just in researches?
It's mostly in research stage at the moment. Of course, the difficulty for human populations is what piece of tissue or what sample might you use to measure that biomarker, and when can you get that sample? And so the studies that I showed were looking at umbilical cord. And so that might be one piece of tissue that we can get at the time of birth that will give some sort of indication of prenatal life. But no, it's mostly at the research stage at the moment. And we'd be looking at risk factors in the whole rather than measuring specific things. We can't do that so much yet.
Okay, thank you.